So now that you know a little bit about virus biology, how does that biology help us understand the diseases that viruses cause? So the first thing that we'll talk about is influenza. So as you'll remember, influenza is a virus that causes upper respiratory infections, typically in humans. And we discussed a little bit about how influenza reproduces by hijacking animal cells in part 1. There are 4 different types of influenza viruses. Influenza A, B, C, and D. Only influenza A, B, and C infect humans, and only influenza A causes flu pandemics. So by the end of this lecture, you should be able to explain why for the exam. So the first important part of influenza biology is the attachment to the receptor. Influenza viruses attach to the cell by having the hemagglutinin spike protein bind to the host cell glycoprotein containing carbohydrate sialic acid. So that attachment triggers the process receptor-mediated endocytosis of the virus, allowing it into the cell. Sialic acid is an attractive receptor for influenza because it's found on the membrane proteins of many animal cells, making this a good site for the virus to attach. Virus receptor interactions are important and specific. The host range or tropism of a virus is usually determined by whether or not that host cell has a matching receptor. Receptor proteins aren't just on the host cell to allow the virus in, they have important functions for the cell itself. The virus hijacks these proteins to gain access, but sialic acid glycoproteins are actually there for cell-cell adhesion in animals. They allow those respiratory epithelial cells to stick together. Another important aspect of influenza biology is replication of the RNA genome. So remember from our discussion of the central dogma that cells have genomes made of DNA. They have enzymes to replicate those DNA genomes. They have enzymes to express genes from that DNA into RNA polymerases. And they have ribosomes in order to translate those RNAs into proteins. There's no enzyme in the cell to make a copy of RNA from RNA. So RNA viruses need to bring along a viral enzyme, an RNA-dependent RNA polymerase, or RDRP, because that is an enzyme that's able to replicate RNA from an RNA. HIV is another RNA virus, but it replicates slightly differently from influenza. HIV is a retrovirus. HIV attaches to and penetrates a cell similar to influenza, receptor-mediated endocytosis. HIV spike protein, GP120, binds to 2 receptors on the cell, the primary receptor, CD4, and a co-receptor, usually CCR5. These are 2 proteins that are found in the membranes of human immune cells. So the type of cells that HIV is tropic for. So once inside the cell, a viral enzyme called reverse transcriptase replicates HIV's RNA genome into DNA. So the RNA genome is made into a copy of DNA. And if you recall, this is opposite the way things work in regular cells. The reason this happens is that so another HIV enzyme called integrase can cut and paste that DNA copy of the HIV genome into the host cell DNA. So the HIV genome actually becomes a part of the host cell chromosome. The viral DNA in the host cell, we call that the provirus, is then transcribed and translated to make new HIV virions. So HIV integrates in the cell and becomes a persistent infection. So many viruses make people sick, but they're resolved within a short period of time. Persistent infections happen when the virus is not completely cleared from the host. It can happen a couple different ways. One, latent infections where the virus remains dormant inside the host for a long period of time, and also chronic infections where low levels, usually, of virus continue to reproduce for long periods. So, HIV infects CD4 positive T cells, and those are immune cells that are only usually active in response to specific immune stimuli. When those cells go dormant, then the provirus can persist inside of those cells for decades without producing viruses. So the problem with this is that neither the human immune system nor HIV medications can clear those latently infected T cells, and they can go back to reproducing viruses at a later time. So this makes HIV extremely difficult to cure. Varicella zoster virus is another virus that goes latent. Varicella zoster causes chickenpox, which usually unvaccinated children get. The virus infects skin epithelial cells, and then for reasons that we don't understand, the viruses in those cells move into nerve ganglia cells and go dormant for years. The virus can reactivate in adulthood and cause a really painful shingles infection. A third example of viral latency are oncoviruses. These are viruses that cause cancer. Cancer is a different disease. What is cancer? So cancer is caused by the uncontrolled growth of cells. So if you can imagine an animal that's made of trillions of cells, this growth of those cells has to be controlled very carefully to make those cells work together. And cell growth in animals is controlled by two types of genes. Oncogenes, which are genes that induce cells to grow, so these are normally expressed in growing cells, and tumor suppressor genes that stop cells from growing. These are normally only expressed in resting or differentiated cells. And cancer is caused by oncogenes being turned on when they're not supposed to be, or tumor suppressor genes being turned off when they're supposed to be on. The vast majority of cancer is caused by a combination of these, multiple mutations that lead to the uncontrolled proliferation of cells. Human papillomavirus, or HPV, is a DNA virus that's tropic for human epithelial cells. HPV infections cause warts. HPV infects basal epithelial cells, but it's released from keratinocytes. So, in order to produce virus, those host cells have to grow. HPV gets the cells to do this using two virus genes, E6 and E7. These genes disable human genes P53 and PRB, which are two major human tumor suppressors. So by turning these genes off, the virus induces the skin cells to grow uncontrollably, creating warts and producing lots of virus. And then in some cases, the virus DNA becomes permanently integrated, and this can lead those warts to turn cancerous. HPV causes over 90% of cervical cancer, but also anal, penile, and mouth cancer. You should get vaccinated because the HPV vaccine isn't just a vaccine against genital warts, it's a vaccine against cancer. This figure really nicely illustrates how HPV leads to cancer. So HPV virions enter into those basal epithelial cells at the lower level of the epithelium through microabrasions that usually happen only through sexual intercourse. Then, once inside those cells, those E6 and E7 genes get those basal epithelial cells to grow and differentiate into keratinocytes, producing viruses and warts. 90% of those infections heal within 2 years, but in a small number of cases, the HPV DNA becomes permanently integrated into the cells, and that can lead to the development of cancerous tumors. So Harold Zurhausen was awarded the 2008 Nobel Prize in Physiology and Medicine for showing that the vast majority of cervical cancers contained HPV DNA. So we had diagnosed HPV infections in precancerous cells using a microbiology stain called the Pap smear. So Pap smear is made a lot like the cheek cell smear that you did in lab. But those cells are stained with a blue dye that binds to DNA. So chorocytes are cells that stain darker blue because they have more DNA from the replicating HPV virions inside the nucleus. So which of these cells do you think are chorocytes? I'll give you a hint. They're the cells with the larger nucleus. The term that we use to describe these differences in cellular appearance caused by the viral infection, I'll remind you, are cytopathic effects, or CPEs. Another important aspect of viral biology that infects viral disease is the reason that we need a new flu vaccine every year. So that's kind of obnoxious. Why is that? So the reason is that influenza hemagglutinin and neuraminidase, these are the spike proteins that are critical for attaching and releasing from host cells. These are the structures of the protein that determine what host the virus can infect. The spike proteins are also the most common antigens for the immune system. They're on the outside of the virus, so they're the things that the immune system typically recognizes in order to get rid of influenza virus. So what specific structures those H and N proteins have determines what strains of influenza someone has immunity to. So the RNA-dependent RNA polymerase that replicates influenza doesn't replicate the virus genome as well as DNA polymerase replicates cell genomes. RDRP makes lots of mistakes. Those RDRP errors mean that the sequences of those H and N proteins mutate very frequently. So this often isn't always good for influenza, but it means that the influenza strain that predominates one year will probably be a little bit different from the one that predominates the next year. Sometimes this will make the influenza viruses less infectious. Sometimes it'll make them more, and usually it means that people who were sick one year won't be completely immune to the virus the next. This process of mutation in H and N we call antigenic drift. The other reason that flu rapidly mutates is a process called antigenic shift. So, influenza has 8 RNA genome segments. So, when an influenza virus infects a host cell, rarely, but sometimes, another influenza virus that has different genes can also infect that same cell. Now, those two influenza viruses have genomes that are similar enough that when new viruses are created inside of the cells, those genomes can recombine. Now, because of antigenic drift, there are lots of different variants of the H and N genomes in nature. There are 18 different variants of hemagglutinin and 11 variants of neuraminidase. And so if you see the abbreviations H1N1 or H5N1, those are different versions of those H and N spike proteins. So if those two influenza strains infect the same cell, those 8 RNA chromosomes can get recombined to create an entirely new virus, one that has a mixture of the two viruses that infected the host cell. This can create a dramatic change in those H and N proteins, create a new virus that people don't have much existing immunity to at all. So, how does antigenic shift cause flu pandemics? Influenza A is a virus that infects several different animals, including birds, pigs, and people. So bird influenza A usually doesn't infect people directly, but bird influenza A can infect pigs. Human strains can infect pigs too. Bird strains and human strains can undergo antigenic shift in pigs, creating new influenza A strains that humans have little immunity against. This is likely how the 2009 H1N1 pandemic strain arose. Influenza B and C infect humans, but not very many other animals. These viruses cause serious disease in people, but pandemic flu has only been caused by influenza A. Why don't B and C cause pandemics? Make sure you can answer that question for the exam.